For a long time, I've worked on lethal degenerative brain diseases. Many of you probably know someone with Alzheimer's, Parkinson's, or some other neurodegenerative disease. If you're a soldier or an athlete, you're probably rightfully concerned about CTE, or chronic traumatic encephalopathy, that is caused by various types of brain trauma and can strike you down in the prime of your life. If you're a consumer or a producer of beef, you're probably well aware of the BSE, or the mad cow disease epidemic that peaked in the late 90s and killed at least hundreds of thousands of cattle and a couple hundred people who ate the tainted beef. If you're a hunter or a consumer of deer, elk, or other cervid species, you may well be concerned about chronic wasting disease, or CWD, which is running rampant in populations in North America, Northern Europe, and Korea. And you may well have dropped off a part of your kill at a checkpoint for CWD testing. So what do these diseases have in common? Well, they're all caused by the corruption of our very own proteins. And so far, we have very little idea of how to stop this process once it really gets going in the brain. So figuring out how to diagnose, treat, and even prevent these diseases in the first place is one of the more consequential frontiers in biology and medicine right now. Why am I being alarmist about this? Well, if you just think of the two most common of these diseases, Alzheimer's disease and Parkinson's disease first, there are 6.5 million Alzheimer's cases in the US alone, including about 11% of people over the age of 65. And the prevalence of Parkinson's disease is about a quarter of that of Alzheimer's disease. Collectively, these and other diseases represent huge and increasing burdens on society unless we can figure out how these diseases work and what we might be able to do to mitigate their effects. Hopefully, ongoing research will head off the worst of what's to come with these diseases. Already, we've made some progress, have a long way to go, but some basic themes about these diseases are emerging. So you may know that proteins are long chains of amino acids, kind of like beads on a string, with each bead having a little bit different chemical property. And there's tens of thousands of different proteins in our tissues, acting as little machines and structural elements. But in order to function properly, proteins have to fold up, usually, most proteins do, into very discrete structures. But sometimes problems in this folding process can occur, exposing sticky surfaces and allowing proteins to clump together and accumulate in our cells. Usually we have pretty effective quality control mechanisms that can recognize this sort of cellular garbage and do something about it. But sometimes these quality control mechanisms can be overwhelmed, allowing these abnormal protein clumps to accumulate to the point where it's able to cause, especially in the brain, cells to die and malfunction. So in fact, if you look at the brain of an Alzheimer's patient, you can see that all of this cell death that happens during the disease process results in a pretty dramatic shrinkage of the brain relative to a healthy brain. We're trying to learn how and why this happens during the disease process, not only in Alzheimer's disease, but in all these other different diseases that may have different mechanisms. And I can tell you right from the start that it's a very complicated set of circumstances to figure out. So one of the main aspects of these diseases is that these abnormal protein clumps, once they get going, can even grow and replicate, in some cases like infectious agents. So they grow by grabbing onto normal forms of the same protein and adding it on the end of what's usually sort of a thread-like deposit. And then little bits of these threads can sometimes break off and move to other cells, to other tissues, and sometimes to other individuals to spread this whole insidious disease process. In fact, sometimes some of these types of abnormal protein clusters are so good at this that we've looked at and know about anyway, is that 1 milligram, or about a pinhead's worth of this material, can contain about 1 billion lethal doses if injected into the brains of a susceptible host. So such infectious protein clusters are called prions, and the classical prion diseases include, that we've studied a lot so far, include BSE or mad cow disease, chronic wasting disease, and their human counterparts like Creutzfeldt-Jakob disease. Making matters worse, the known infectious prion structures anyway are typically unusually resistant to treatments that are frequently used in clinical settings or in food processing or even cooking to kill other types of pathogens like viruses, bacteria, and fungi. And they also can survive, or at least some of them, for years in the environment. It's probably the most contagious of these sort of protein corruption diseases or proteinopathies is chronic wasting disease. And CWD-infected animals can shed these infectious prions into the environment in their urine, their feces, fluids, or even from their carcasses when they die and scavengers tear them apart and spread them in the environment, where again they can last for years until perhaps they're picked up by another animal to initiate another infection. But before we get too worried about this, got to realize that the basic hygiene of wild animals is much worse than it is for humans. And so far, there's very little or no evidence that I know of any sort of casual transmission between humans the way we see running rampant in the case of CWD and these cervid animals. Then there's the issue of the extent to which CWD and cervids can actually pass into humans who are hunting and eating these animals or into other livestock species that we also eat. But again, so far, there's no solid evidence that CWD can pass from cervids into humans at this point. Our experience with the transmissibility of these classical prion diseases raises the issue to the extent, if any, these other much more common proteinopathies, such as Alzheimer's and Parkinson's disease, might similarly be transmitted between humans. There are laboratory experiments showing that Alzheimer's, Parkinson's, and some other of these diseases can be transmitted, in a sense, from humans into susceptible laboratory animals. So the big question right now, and it's the extent to which this might really happen, this kind of transmission between humans in the real world. The vast majority of these sort of the common proteinopathies in humans arise spontaneously and are not due to infections from some prion-like infectious pathogen. So just as important as how these corrupted protein clusters grow is how is it that they cause problems? How are they toxic? And there are a lot of ways we can think of that the accumulation of this sort of protein crud within cells or around cells can interfere with cellular function, especially in the brain, where the neurons or the nerve cells there have to make all of these intricate signaling connections. And these cells are particularly vulnerable to being poisoned and killed. But despite the fact that we can envision all kinds of ways that these proteins could be toxic, we're still unsure about the most relevant ways that they are toxic and what we might be able to do about them. So another big challenge in dealing with these diseases is the ability to diagnose these diseases accurately, especially early in the disease process, when the clinical signs of these diseases can be variable and overlapping between multiple proteinopathies. In fact, you may have a friend or family member who is bounced from test to test or doctor to doctor, sometimes for years before the doctors get a fairly good idea of what's going on. And unfortunately, definitive diagnosis of many of these diseases requires analysis of the brain tissue after the patient had died, which of course is too little, too late. And it's certainly hard for doctors to deal with any problem when they don't know its cause. Fortunately, we and other labs around the world are developing much more accurate diagnostic tests for some proteinopathies that are working really well so far for prion diseases, Parkinson's disease, and diseases closely related to Parkinson's. These tests are based on the ultrasensitive detection of the protein clumps themselves that cause the disease. In fact, these tests can be so sensitive that you can take, again, a milligram or about a pinhead size amount of contaminated brain tissue, for instance, dilute it into an Olympic swimming pool and take a similarly small sized volume out of that swimming pool and still get a positive test in some of these reactions that we run. And we in other labs are working hard, many other labs are working hard to expand the panel of these kinds of tests to many other types of proteinopathies, so that when a patient first comes into the clinic with their clinical signs, the doctors will be able to tell pretty quickly what their underlying problem or problems are. Being able to do this early, before much damage has been done, is really crucial in being able, in potentiating our ability to develop and implement therapeutic strategies. Some of the therapeutic strategies that people around the world are attempting to develop are targeting the conversion of the normal protein to this corrupted form, and then they're also targeting the toxicity of the corrupted forms that may have already accumulated in the patient, and also ways of promoting the body's ability to clear such aggregates after they've occurred. But unfortunately, we're still failing pretty miserably in developing actual drugs that work actually in human patients that affect the underlying disease process, but not for lack of trying. You may have heard about many failed clinical trials of drugs that are meant to treat Alzheimer's disease. But there are some glimmers of hope. I mean, you might have read very recently about a new drug called lecanemab that does, in pretty large clinical trials, appear to at least moderately reduce the rate of cognitive decline in early Alzheimer's patients. It's hardly a cure, but it seems to be progress in the right direction. On another front, we and our collaborators here at RML have found that a completely different kind of drug can really substantially prolong the survival time of prion-infected mice. What can you do to help? Well, one thing would be to try to stay informed and support the efforts of governments, universities, foundations, and we all rely very heavily on basic information, on tissue specimens, and so forth from the patients and their families who've very generously donated these specimens. And getting these precious specimens is often one of the slow steps in the work that we do. But unfortunately, there's no escaping the fact that clinical research and drug development is hugely expensive and has many blind alleys associated with it. But after all, research is our attempt to understand what we don't already know. So it's not like clockwork. We come up with ideas, hypotheses, and then we go to the lab or in the clinic and try to shoot those ideas down or confirm them and build on them. Real, tangible progress is being made in this war. And what we need to do at this point is keep at it until we're best able to maximize people's functionality and quality of life for as long as possible. And of course, this not only benefits the patient, but all of the people in their world around them. Thank you for listening, and then thank again the many patients and their families who are providing us with specimens and information that we are trying to use to do better in treating these diseases.